This is USMLE QA, USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: what is the direction of DNA replication? Answer: DNA replication occurs in 5 feet greater than 3 feet direction in continuous and discontinuous fashion. Question: what is the nature of DNA replication? Answer: DNA replication is semiconservative, more complex in eukaryotes than in prokaryotes, but shares analogous enzymes. Question: what is the origin of replication? Answer: the origin of replication is a particular consensus sequence in genome where DNA replication begins. May be single or multiple. At-rich sequences are found in promoters and origins of replication. Question: what is replication fork? Answer: replication fork is a Y-shaped region along DNA template where leading and lagging strands are synthesized. Question: what is helicase? Answer: helicase unwinds DNA template at replication fork. Helicase halves DNA, deficient in Bloom syndrome. Question: what is the role of helicase in DNA replication? Answer: the role of helicase is to unwind the DNA template at the replication fork in order to allow for the leading and lagging strands to be synthesized. Question: can a deficiency in helicase cause any genetic disorders? Answer: yes, a deficiency in helicase can cause a disorder called Bloom syndrome, which is caused by a mutation in the BLM gene. Question: how is DNA replication more complex in eukaryotes than in prokaryotes? Answer: DNA replication is more complex in eukaryotes than in prokaryotes because eukaryotic cells have multiple origins of replication and also have a more elaborate mechanism for replicating their larger and more complex genomes. Question: what are at-rich sequences and where are they found in the genome? Answer: at-rich sequences are regions of DNA that contain a high proportion of the nucleotides adenine and thymine. They are found in promoters and origins of replication in the genome. Question: what is the significance of the TATA box region in DNA replication? Answer: the TATA box region is an at-rich sequence that is typically found in the promoter regions of genes, and it is also an important regulatory element for the initiation of DNA replication. Question: what is the function of leading and lagging strands during DNA replication? Answer: during DNA replication, the leading strand is synthesized in the 5 to 3 direction, while the lagging strand is synthesized in the opposite direction, in short fragments known as Okazaki fragments. The leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously. Question: what is the difference between continuous and discontinuous DNA replication? Answer: continuous replication refers to the synthesis of one strand of DNA in a unidirectional manner, while discontinuous replication refers to the synthesis of both strands of DNA in a bidirectional manner, with short fragments known as Okazaki fragments. Question: how does DNA replication differ in prokaryotes and eukaryotes? Answer: DNA replication in prokaryotes is relatively simple compared to eukaryotes. Prokaryotes have a single origin of replication and a smaller genome, while eukaryotes have multiple origins of replication and a more complex mechanism for replicating their larger genomes. Question: how does the process of DNA replication ensure genetic stability? Answer: the process of DNA replication ensures genetic stability by accurately copying the genetic information from one generation to the next. The semiconservative nature of DNA replication, in which each strand of the original DNA molecule serves as a template for the synthesis of a new complementary strand, ensures that the genetic information is passed on accurately. Question: can mutations occur during DNA replication? Answer: yes, mutations can occur during DNA replication due to errors in the replication process or exposure to environmental factors such as radiation or chemicals. These mutations can lead to changes in the genetic makeup of an organism and can have varying effects on its phenotype.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses, which are structured to follow along with the first aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.